All right, so here we go. So the genetic information that makes you came from mom and dad. How many chromosomes do you have in each cell? 46, right? 23 pairs. So that means one set came from mom, one set came from dad. Everybody good with that? Okay. So the information on your genes, that biological information gets not only passed down to the different cells, right? Because you start at one cell. That cell no longer exists. It's gone a long time ago. They say that nearly every 7 years you replace most of your cells. So truly you're not who you were 7, 8 years ago, right? So how does that information move forward from generation to generation? And that's the central dogma. So the big deal here is that the information that is on DNA, so we're looking at information, is on DNA, goes to RNA, and then to a protein. And this is what happens within the cell. So that information then gets passed on through gametes. And we make gametes. What's the process by which we make the gametes? Meiosis or meiosis. It doesn't matter how you pronounce it. Okay? So our central dogma is this big thing right here. And so this information has to be passed on to the next generation so that that information can then go on to that next generation. We create the proteins and we still exist. Is everybody okay with that? That's central dogma. Okay? All right. So we will do, because I don't think we've done protein synthesis yet, correct? Yeah, we will do the protein synthesis piece a little later. What I'm focusing on this time is past meiosis and how the genetics piece works. All right, are we okay? Okay. So if you look on the study guide, the first thing it says there, explain how shared conserved fundamental processes and features support the concept of a common ancestry. We're going to get to more detail about that a little later, but this is how we have that common ancestry, because if natural selection is correct, if evolution is correct, then the idea is that you are a product of a mom and a dad who are a product of your grandma and grandpa, and then there's 4 of those, right? And then you keep going back, and so we are all a product of this central dogma and the information then being passed on. Okay? The same processes that work in bacteria, lower life forms, plants, are the same processes that work with us. And we'll talk more about that when we get to heredity. All right, so have you heard of Gregor Mendel? Math teacher, Gregor Mendel. You don't, yeah, the monk. You don't really need to know all that he did, but I would be able to recognize his name, Mendel. Okay, make sure you can recognize his name. He's considered the father of genetics. He did a lot of things with pea plants, so you might get a question to ask about pea plants. But we're going to do what we call Mendelian genetics today. And he had more than these 2 rules, but these are the 2 rules that AP wants you to know. And they're on there under that second paragraph. And that's the law of segregation and the law of independent assortment. So we're going to do those 2 things next. And then I think we'll talk about, see, I've got down here in my list, fertilization, and then we'll talk about actually how to do these things. So the law of segregation. I wrote the word down so I would not mess it up. This basically means that you get 1 allele for each gene from each parent. Do I need to explain? Yes? Okay. So mom, dad, they have all these chromosomes. I'm just going to focus on 1 chromosome from each, okay? Now they're paired, right? So there's the 2 chromosomes, 2 from mom, 2 from dad. And 1 of them will come in. These are the same chromosome number, right? So if there's 23 pair, maybe this is chromosome 1 or chromosome 5. And 1 of them will come in from mom. So maybe mom has, W and little w, and dad's got big W's and big W's. Well, that means dad's only going to give a big W, but mom may give a little w, or mom may give a big W. The law of segregation says you're going to get one of these from each of the parents, and it's random which one you get. That's all that is. I think the reading has a, see, I can't get to the reading at the moment. Hold on here. I think the reading it's in chapter, it's 12.3. It's the last reading. It's very short. It has a definition in there for you if you'd like to have a final definition. But basically, it means you get one allele from each parent. Okay? So you're going to get one set of chromosomes for each parent. And then how those things come to the fertilized egg, is random. That's segregation. How they segregate is random. How these come apart is random. Okay? And if you think about all the genes that would make you up, that's a heck of a lot of genes to randomize. Do I need to go into more detail on that? No? Okay. I did post, I had a 2022 lecture of this, and I did post that also underneath the resources if you wanted to see it. That's the law of segregation. Okay? The law of independent assortment is not, it is a good law, but we now know that it's not 100% true. Okay? But when they ask you about the law of independent assortment, I actually wrote a definition down that I made up that I think is a little better. Genes do not influence each other. So genes do not influence
You say that they're independent of each other?
They are independent of each other. That's actually the next thing on my little definition. The genes are independent of each other. The only exception is linkage. So if I had a chromosome like chromosome 1 here, and it had, the A allele on it, and I had chromosome 2 and it had like the E allele on it, whatever that makes, they are going to independently assort. Are we okay? In other words, big A could go with the big E, a big A could go with the little e, a little A could go with the big E, a little A could go with the big E. It's just they separate out. They're not dependent. How that gets divided is not dependent on how that one gets divided. Independently, they separate. So far, so good? We're going to talk about linkage a little bit, and we'll talk more about linkage next time. This is really non-Mendelian genetics. That's our next class. But what if the chromosome had an A and a B gene on it, like all our chromosomes do? A and B cannot assort independently because they're on the same stinking chromosome. Okay? So, genes do not influence each other on different chromosomes. So that's the little add-in that you want to put on the end of that. And it's this, linkage is the thing, and we'll deal with linkage later. Okay? AP, multiple choice, test this. I've never seen this on a short answer or free response question. It's always been a multiple choice question. They want to know, do you understand? Not can you recall it, but do you understand what this means? Okay? Any questions on any of this? All righty.
So is linkage just if they're on the same chromosome?
Yeah, the genes are located on the same chromosome. And we will have what we call X-linked. You won't probably get Y-linked, and sometimes it's referred to as sex-linked genes. Any questions here? Okay, I'm gonna erase this. Make sure the thing is still rolling. Okay, good. All righty. So in some ways, I think it's good that you may not have been exposed to things. In some ways, it's bad. Let's talk about dominance and recessive. I disagree with a lot of especially biology teachers that teach regular biology or life science teachers in the middle school. And I think it's because they feel like you can't handle the truth. But this is the truth that I'm getting ready to tell you, and it's the way AP looks at it. When you hear the word dominance, right? Dominance, you think of someone that's stronger, something that's bigger or better, okay? Not the case. And a lot of biology teachers teach it that way, but that's not the case. I'm going to change the definition for you. Dominance means the gene is expressed. Central dogma works. DNA, that code, we make the protein, we express the gene. That's what dominance means. The gene is expressed. Recessive, it's hard to say it's not expressed, but I'm going to put on here not expressed or non-functional or partially functional. I know that's a lot to kind of grasp. Today we'll deal mostly with dominance and recessive being it. But there is a thing called incomplete dominance. And the best way to explain this is give you an example. If I have red flowers, and that gene, that allele for that gene is dominance. Is dominant. You know what I mean when I say allele, right? Do I need to define that?
Yeah.
Okay. You okay?
Yeah.
Okay. You guys okay? I made the assumption there and I apologize. And let's say that white flowers are recessive. Okay? So that means if my genes, I get 2 copies, right? So that means this would be big R and big R. The capital letter indicates dominance. This one would be little r, little r. But there is a condition where we have a partially functional recessive, and we get what's called incomplete dominance. And so we may get pink flowers because this is the heterozygote, and that's big R and little r. So the dominant gets expressed, but because we have a partial functionality here, we're getting a mixture of those 2 colors. Okay?
Is it all up to probability on whether or not you get it? Like, is it all?
Right. So how these things fall out is, for right now, all due to random chance. Okay? So are we okay with this? Okay. Do you remember what heterozygote means? That's right. So this one would not be heterozygous, this would be homozygous. Zygous, this has to do with the embryo not doing it quite right, and this means same. So it's not so much the same embryo, it's the same allele. Okay? Same allele, same allele. Everybody good with this? All righty. And then there's another option. I'm running out of room. Is it okay if I erase? Okay. I just want to be sure that you can see. I'm going to erase this one up here. And this is codominance. Suffix prefix quiz. What does co mean?
Together.
Together or with. So in this case, with our red flowers, and I'm just going to erase incomplete so it won't confuse any. So with our red flowers, so that allele may be big R. White flower gene, the allele may be big W. So far, so good. So the homozygous, a homozygous red would be what? And a homozygous white so what would the heterozygous allele genotype, this is called the genotype, what's on the genes, the heterozygous would be what? Big R and big W. So it would be splotchy red and white. Phenotype is always what is expressed. It's the physical look of it, is how I usually say that. Okay? So this isn't like zebras. First of all, I want to make sure that's really clear because that's a whole separate thing. That's multiple genes and multiple alleles.
Or like dominance and for dominant and recessive traits.
Mm-hmm.
Is there a different probability on whether or not which one's like going to happen?
Nope. There is not. There is not. I'll go ahead and say this, but you might want to just kind of put it in the back of your head for now. The genetics doesn't necessarily indicate or be exactly what happens. There is this whole argument of nature versus nurture. How much is genetics and how much is the environment that the person is in, the organism is in. Okay? So we're going to say that for now, right now, we're going to say that nature rules everything. If it's there, it's dominant, it's expressed, period, end of story. Okay? AP usually doesn't hit that level of stuff with you all, this epigenetics piece, but there is there. So, like your hair color has 6 different genes. I think your height is 8, and they all work in combination. But nutrition plays a big part of that. Where you live plays a big part of that, because of how much exposure you get to the sun. All of these things, your environment plays into that. So it's a combination of the two. Does that make sense? But there isn't any special rules. Oh, it's dominant, so it's going to do. No, there's no specialness there. Okay?
So there's that equal chance of it just being dominant and then being incomplete dominant and complete dominant?
So that's a little different because there is those processes. You have straight dominance, right? Dominance, recessive, straight, everything's done. You have incomplete dominance, okay? That's another pathway that can be done. You have codominance, that's another pathway. But those are all specific to the actual function of how that gene works. Does that make sense? It's not something that's straight dominant can suddenly become incomplete dominance. There would have to be some major mutation to get that to happen. Is that answering it?
So if you had red and white flowers, would there be an equal chance? So if you had homozygous red, so okay. I think what I'm more confused with is like the recessive trait being in the gene. So like, what's the difference between a white flower that's R, like capital R, capital, and then a white flower that's lowercase r, lowercase r?
So it's how that gene gets read. Okay, so it's the recipe. It's built in. It's the information that is already there in the DNA. Okay, and so on a test or on a worksheet, if it's not straight dominance, they will say this and this show incomplete dominance. And so now you know that the mechanism by which that one works is incomplete dominance. And that's encoded in the original DNA sequence. Okay, I may not be understanding 100% your question. And you'll get that information up front. In other words, I won't give you one, AP won't give you one without saying incomplete dominance or codominance. It's rare they do codominance, but they do it sometimes. Mostly it's incomplete dominance and it's multiple alleles and sex-linked or X-linked. Does that make sense? Okay. And if you think of a better way to ask the question, just ask. Yes, ma'am.
Can you re-explain dominance and resistance? Like, what do you mean by if we express like that?
Sure. And this, even though we jumped to genetics instead of doing protein synthesis, I think protein synthesis actually does follow it on their list. So you have DNA sequence. I won't get you all the... So this is the sequence of bases on a piece of DNA. Do you remember that stuff? The nucleotides, adenine, guanine, cytosine, thymine. Okay. So that's what this is. And they're all kind of hooked up together on a strand of DNA. So far, so good? In the process of making the protein that comes off of that, that's the actual expression. And so that process, we would get an mRNA that would come in. So there would be your RNA piece. And so that would come in. This would be U, C, U, A, G, C, G, U, A, right? This sequence here is a code for a protein or something similar, but usually it's a protein. We'll stick with proteins in here because it's 99.9% it's a protein, or it makes a protein that does another product anyway. It doesn't matter. So then this goes out to the cytoplasm and the protein is actually made off of this sequence here. So far, so good? That's expression. But for recessive, let's say these two turn to A's. Now we have a different code. It may still make the protein, but instead of the protein being what it's supposed to look like.
So essentially recessive is a type of mutation?
No. No. It may be that this did happen and it was a mutation. It may be that there's several proteins that work together, so instead of it just being one protein, it's a couple of proteins, and one of them is messed up, so it doesn't bind as well to where it needs to bind, but it still binds. So it's only partially expressed. And then some of it is straight recessive. It makes the protein, but this thing can't do its job at all. And so we don't get it. So dominance, man, we made it. It's perfect. It's great. It's working. It's been expressed. Okay. Recessive, you can still have something expressed. I don't like saying not expressed, but it could be expressed and it just is crap. It doesn't work. It can make a disabled protein. Now there is some, and we haven't got to it yet, but there is some things that will literally shut it down to where this won't even work. And if we go back to cell signaling, there are ligands that can cause a cell signaling that switch off a section of DNA. So even though it may be expressed, the environment may keep it from being expressed, so we no longer have that protein being made. That's why I'm saying it's more than just the genetics I'm showing you. It's going to be more later. We just aren't doing that right now. We're just going to say, dominance, man, we express it. We get what we need. Product comes out. You have black hair, right? Does that make more sense? These are really good questions.
How common are things like codominance and incomplete dominance?
I think they're more common. I do not know the correct answer to that, but I think they're more common than when I went through school in high school, because we were thinking it was a lot of this. Amazing. It's amazing that Gregor Mendel picked 7 traits on a pea plant that showed straight dominance. Absolutely amazing. And it's believed when we look at what he's done that he literally knew enough statistics. He was a mathematician, so he knew statistics, manipulated his data to show that this worked. And it works. Most of the time what I see on an AP test, and most of the time what I see on the others, it's not simple, straightforward dominance. It's usually incomplete dominance or multiple alleles or sex-linked or X-linked traits that they look at. Yes, ma'am.
Can you explain the excellent trait?
That's where I'm headed. I'm headed to that. Yeah. Any other questions? These are really good questions. Ones I would not have expected. Yes, ma'am.
I have a question about like the horizon thing, shutting down like the different.
Mm-hmm. That would be the environment affecting the genetics.
So like you know how sometimes like certain like environmental factors can like hinder like the development of like embryo and stuff. Is it because like the ligands are shutting down the different?
It can be, or switching them on and they shouldn't be switched on. So remember, fertilization, right? Fertilization is egg, sperm come together and a zygote, a single cell is formed, okay? After that, we do mitosis to do its thing, correct? Well, if all the instructions for you are on that one cell, some of those need to be switched off, right? You don't want to start puberty inside mom, right? I mean, think about it. So things have to be switched on in a correct order. There is some genes that actually control what gets switched on and off. So if you are, if there is a substance, and I don't know if alcohol does this, but it can cause problems with the development of the baby inside. So there are substances that can get down onto that baby and can cause a disruption in how that order gets shifted or cause mutations in that. And again, that's all going to be environmental, not this straight genetics piece, okay? And that's what they're going to do. They're literally, what you guys are asking, they're literally going to go back and grab cell signaling and pull that into genetics. If you noticed when we took that test, there were, like it said, B and C or A and D or whatever. Those other two probably were genetics questions. Yeah, it's really complex stuff. Anything else? These are good questions. I forgot to say what your question was, so they may not have heard it anyway. Are we okay? Okay. Let's get down to the brass tacks of it. Let's get down to the nitty-gritty piece. See how much of this I can do. Let me make sure I'm in. Alrighty. You don't necessarily need to write this down if you've got it. Okay? I'm going to tell you there are, let's go back to the flowers. It's complete dominance. And we have a red heterozygous crossed with a white recessive. Or right, I'll use the word homozygous. If they give you the letter, you must use their letter. Got it? Do not make up letters. If they give you a letter, do not make up letters. You will get it wrong. Period. They won't even look at the rest of it. They didn't give me any letters here in this problem I just made up. So we're going to use R because usually, so you can determine it's complete dominance for red. So usually the capital letter is, the letter you use is the one that's dominant. So far, so good? So step one is always going to be to put your parent's genotype. Here's the deal. These 4 steps that I wrote over here for answering these, you have to kind of manipulate them a little bit. They're about 80% correct all the time because sometimes they'll give you the parents. Sometimes they will give you part of a parent, but then they'll tell you about the parents' parents, the grandparents. And then you have to deduce from the grandparents what the parents' genotype is to figure out what the kids' genotypes are. Does that make sense? So the first step is I always say, if it's appropriate, you always do this. So if it's heterozygous, this means the parent genotype is going to be what for the red? Big R and little r, right? And then for the white, it'll be Complete dominance. Yeah, when they do something that's codominance or codominance, they will give you they'll give you the letters. So it's complete dominance, so you're going to have to use. So far, so good? You remember this? Step 2 is always figuring out what the gametes are. In other words, this parent can give either a big R or a little r. If you're not comfortable yet, I would strongly suggest that you write them down. Big R and little r. Everybody good? Go ahead.
When we see a homozygous, do we automatically assume that it's little r and little r?
Well, we chose the R for our thing, but if you see it's homozygous and they tell you that red is dominant, then you would have to do that way, right? But if it said white was a dominant, then this one would be big R and big R, correct? Okay. Now there are some teachers here. So first of all, what's my type of gamete that I can have here? Yeah, I can only have a little r gamete here, right? Next thing you do is you can do the Punnett square. I'm going to tell you at this point in your existence, by the time you take a genetics test, by the time we get to the AP test, you should be able to tell me without writing anything down what the percentage for the genotypes and the phenotypes are for this. Okay? The Punnett square is a little bit of a crutch. So some teachers will tell you to do that. I wouldn't. It's a waste of time. All the Punnett square is to show how the distribution of the alleles occur. So you could do that, and I'll do it real quick here. So for my genotype, I get 2 out of a possible 4. I shouldn't say never. For the worksheet, do not leave a fraction. Okay, go ahead and do the math. So that would be the genotype here, 2 out of 4 are big R, little r, which is 50%. So far, so good? And then on the bottom, 2 out of the 4, little r, are 50%. Are we good? If I did the Punnett square this way, half of them are genotype big R, little r, half of them are genotype little r. It's the same thing. You get it? Work smarter, not harder.
But that second type only works if it's the heterohomozygous?
Right.
Okay.
Right. I should be able to, at some point, be able to put this on the test or give you this, and you should be able to tell me, without doing any Punnett square, you should be able to tell me what that genotype would be. And let's think about it. Big A goes with one big A, right? But that's the same thing, right? Big B goes with, that's the same thing. Excuse me, yeah. Right? So it's 100% genotype for that. Are we okay? It's making sense? It's math. I got practice problem for your work. I'm sorry, it's going to end up being homework. So that's actually a good question. Let me erase over here and get rid of this. That's a really, really good question. So I'm going to do this. Really, really good question. If I were to do the Punnett square method, I would have only 2 types of gametes here, right? One type of gamete here, right? I would not get a gamete that looks like that. Gamete, not offspring. Okay? Okay? So one of these has to come, and one of these has to come. In fact, I have an option of 1 here times an option of 1. So there should only be 1 gamete. Okay? So that means I'm going to have big A and big B. Same thing. I only have 1 option here and 1 option there. So that means it's going to be this. Now, you would never, ever write it like this. The capital always goes first. You would also never write it like this because they didn't put the genes in that order. Whatever order they gave you, it doesn't matter whether it's alphabetical or not. Whatever order, that's the order you have to put it in. Yes, sir.
Can you elaborate on these 1 and 2?
Yeah, I'm going to run more problems. I'm going to work more problems for you. That's why I said I'm going to just give it to you as homework, and then when you come back next class, if you got issues, we'll work problems. Okay. All right. So far, so good. All right. Now let me do this. You may or may not use it, but there's some people in here. So if your math skills are weak, you know, la 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 la, right? But if I had this, how many gametes would that one make? One there, or excuse me, two there, one there, one there, two there. Those are both capital, one there. 2 times 2 is 4, times 1 is still 4, times 1 is still 4, times 2 is still 8, right? Times 1, so we'd have 8 different gametes. Okay, we good so far? Okay, again, if you're weak in math, ignore what I just said. You won't get a problem that's that many, by the way.
So
I can do a simpler one. Two options, possible 2 options, possible 1 options. This is just probabilities. That's all that is. So far, so good?
So you have like 9 different options for 8 total gametes.
You would have 8 for that parent. You would have 8 different options for gametes.
8 options for gametes.
Right. So that parent could only produce 8 different kinds of gametes, and that's it. Are we all right?
I think the only disconnect is I'm not having this sliding multiplying.
It has to do with the rules of probability. Yeah, it's I think it's the third reading. The readings were divided up really small. So like the third reading is like maybe a page. Maybe two screens, maybe 3 at the most. All right, let's work another problem here. Let's work a problem. What do you want? Incomplete dominance, X-linked. You want to do an X-linked? You want to do ABO blood group? That's multiple alleles. Okay. So ABO blood group, if you have A-type blood, you make the A antigen. If you have B-type blood, you make the B antigen. That's supposed to be an A in there. If you make O-type blood, so this is the type, then you make no antigen. So can people be AB blood type?
Yes.
They can. The A antigen is dominant. The B antigen is dominant. The O antigen is recessive. So for the A antigen, we use A. But there's a problem before you write anything down on that part. The B antigen, you use B. But there's a problem here. The O antigen, you use a lowercase I. And here's where the problem is. You've been used to, at least when you first did this, seeing just 2 possibilities. There's 3 possibilities. This one has 3 alleles. This is called multiple alleles. So far, so good? So how do we do it, and why is that I? Well, we picked a letter. In this case, it stands for immunoglobulin, which is basically a big class of different proteins that are involved in the immune system. And so now, instead of this dominant, we have to write it capital I, the A allele. So my gene is the immunoglobulin gene. For B, we can't write it that way. We have to write it capital I in our B allele. But O is recessive, so it stays the little I. Okay?
So I is just
I tells us the gene
the gene. Okay. And
because of the multiple alleles.
and B says
Yeah, so A is still dominant, B is still dominant. So what's the genotype for someone that's AB?
Codominant.
It is codominant, but I asked for the genotype. Capital IA, capital IB. Do you see why?
And it can't be capital like IAB. It has to be capital IA, capital B.
Because you have to state the gene. And if you don't put it in this format, it doesn't matter. It's wrong. It's got to be in this format. This is an international format. I know Mr. Pham, you didn't have him. He does teach not using this letter, but he shows it to them later. When they come to me, when the freshmen do this, I'm going to make sure I go through this again with them, because this is one of the biggest mistakes on this type of problem that I see. Now let's talk about someone who is A. What could they have? There's two possible genotypes. IA, IA, and lowercase I. Remember, each parent gives one allele. There just happens to be 3 different alleles for this gene. But the parents only have 2, and they're going to give you one or the other. Okay? So this means, and please understand, just because they're 2 dominants and it's capital IA, capital IA, doesn't mean they're making twice as much. We're not going anywhere near that. Okay? So this person has an I, a lowercase I. So it has one set of its genes is the recessive, and we call that O. And it's not really O, it actually means null. Yeah, we just call it O. Yeah. All right, what about B then? Based on what you saw, what's the genotypes for B? IB, IB, IBI. Boy, that just sounds like a tongue twister or something you'd have to say in like, for what is it, voice or diction? IB, IB. Okay.
As much as it's like topical, where does like the positives come from? Like O positive.
That is RH. And that stands for rhesus monkey protein. It was one of the first proteins that we discovered that both humans and rhesus monkey that we found outside of the human. And so we named it rhesus monkey. So if you're RH positive, you could be RH positive homozygous. You could be RH positive heterozygous. If you're RH negative, you are RH negative. So when they say you're AB positive, they're talking about 2 different genes that control what the blood looks like phenotypically. All right, let's do the last one. If you are O type, which means you can give blood to any one of these guys, right? Because nothing's being expressed, so nothing reacts here and you don't die a horrible, painful death. Unfortunately, this person cannot receive blood from any of the others. Because as soon as it sees this capital A or capital B, the immune system will attack it and blood clots form. Now they've done it and they put heparin in and a few other drugs, but as far as the genetics go, you have to follow this path. So if someone was an IB, and they married someone that was little I, what's my possible genotypes? You don't even need to do a Punnett square. Look, IB, I. Make sense? Okay, we're running out of time.
O is the universal donor, and then AB is the universal donor.
Yes.
So for IAIA is an option for someone, and then IAI is an option, another option.
They would still both show up as making, because you test for the phenotype, not the genotype.
Okay, but can you have them in the same chromosome? Like, are those just 2 different options for the alleles? Or
There are 2 different options for the alleles, but that means this person received from mom that one, or excuse me, you were talking about this one. The person, this came from mom, This came from dad. Because remember, you get 2 copies. We okay?
So it'd be fine if I put IA, then or IA.
Right. Usually, though, on the test, they usually make it obvious, or you have to reason through. There will be one answer. You know, and remember, if it says best answer, you need to put the best one. You would never write this, though, by the way, as this. I will, and so will AP mark that wrong. You always put the dominant first within the gene. Are we okay? All right, let me do sex-linked real fast. It's technical. Yes, it's a technical format thing. It's exactly it. That's the only reason.
Most of the time, the noticing alleles like that, you do the same thing because those are associated.
And they will give that to you.
Okay.
You will be given that information. Now, for the ABO blood group, they probably won't. They expect you to know that. So you should know what I just did. That's a memorization thing. But the other ones, they will expect you to know it. Okay, so let's talk about X-linked, sometimes called sex-linked. They won't be on the Y, it'll always be on the X. This means that the gene is located on the X. Gene is located on the X chromosome. That's what this means. So far, so good? Remember that gender, male or female, is a trait. Okay, please remember that gender, male or female, is a trait. So far, so good? Okay. So male pattern baldness. I'm going to use the letter B. I think they actually use C on the AP test. So let's talk about male pattern baldness. First of all, to be female, what does your genotype have to be? Has to be XX. Now, if baldness is a trait that is X-linked, that means somewhere on the X is baldness, the gene for baldness. Okay? So this X and this X, this is one chromosome that came from mom. This is one chromosome that came from dad. So far, so good? So maybe mom is heterozygote. Male pattern baldness is recessive. So far, so good? And she marries a beautifully haired man. His genotype for gender is X and Y because he's male. His genotype for male pattern baldness is big B. There's no heterozygote and there's no homozygote. He only gets one allele because males only get one X. So the gene is located on the X chromosome. Okay? So if you go bald, you have to blame your mom because that's the only way you get that gene. You got it? So here, what would my gametes be? X big B. We got to do the gametes. There's our parents. X little b, remember? Okay. I'm going to go ahead and draw the Punnett square here. What would my gametes be for this one? And Y. So this one would be X big B, X big B. This would be X big B, X little b, X big B, Y, X little b, Y. So far, so good? So for male pattern baldness, what percentage of the offspring would have male pattern baldness? 25%. 1 out of the 4. Would any females have male pattern baldness? No. Could there be heterozygote carriers? Yes, we have one heterozygote carrier. You see the kind of questions that can be asked? Because you have to answer the question that's been asked of you. Yes, sir.
Why wouldn't it be X big B and little Y?
Because the Y isn't little.
Oh, that's just boy.
Yeah, and that's just boy. That indicates males. If that gender trait alleles changes, like in birds, you'll see a lot of times Z and W, and I don't remember off the top of my head, but I think ZZ is male and ZW is female.
Yeah, but you said that the X will be the male pattern, or
Right, that's this one.
Isn't will be like not dominant?
Right, male pattern bosness is recessive.
So the guy couldn't carry it, but
Now the guy, see, and here's the thing. Let's just do it this way. Let's talk about Henry VIII. He chopped off ladies' heads because they couldn't make him a son. What can the male, female bring? What gamete can she bring? That's it. That's all she can supply. X female. What can the male bring? So who determines the gender? So when Henry VIII was cutting off people's heads, it was really his fault. And that's a very oversimplification of the process, but that was his fault. Does that kind of help? So because of that, anything that's located on the X chromosome, the male can't pass on to his male sons.
oh, sorry, okay,
You got it?
yeah. I just forgot.
That's why I say, if there's an X-linked chromosome and you've got it, you blame mama because dad had nothing to do with it. Unless you blame him because he didn't assort properly when he was making his sperm. Making sense? Sex-linked. Alrighty, I'm wondering if I should just stop here. There's the Fort method. I think I need to wait on the Fort method. I don't know. How much time do we have left?
5 minutes.
I can do the Fort method in 5 minutes. Do you want to look at it or do you want to just stop here? The Fort method, if you're weak in math, it will bother you. Stop here? Okay, I'll stop here. Because the Fort method, if you're weak in math, but once you get it, it's kind of like algebra. Once you got algebra, it's like, oh my gosh, why didn't we do this from the beginning?